Ah, genetics and inheritance, blood grouping. Welcome, guys. I recorded the video and then now the laptop crashed and then I lost the recording. The whole hour, guys, gone. But okay, we can do this again. Genetics and inheritance, blood grouping. So, guys, with that being said, you know that I'll be fasting or I might exclude other things. So welcome, guys, those who are here, those who are new here, we welcome you guys. Those who are returning students, I also like to thank you guys and welcome you once more. Welcome to my class. My name is Mr. Mpepu, life science teacher. Shoda. Genetics and inheritance, blood grouping. Now we are dealing with blood groupings. Today's date 25 March 2024, time 17 past 12. So I recorded the first video around 9, as now the laptop crashed and then I can't find the video, so we are redoing this again. I'm tired, by the way. So let's move. We are heading to week 2, term 2 2024, annual teaching plans. Maybe it was for the good that I didn't get the last recorded video because I was saying a lot of things there. So week 2, and then week 2 we are going to do a blood grouping, guys. So blood groupings, you can see that we are doing blood groupings before dihybrid crosses, guys. There's a more of understanding here. I think you will understand more blood grouping if it was here on top based compared to this. It's not my thing. It's an ATP, and even in examination guidelines, it comes blood grouping. It comes before dihybrid crosses. So here it's a new thing, 2023-2024 annual. So maybe they did a revision and found out, or if we do blood grouping, the last part, learners do understand more when we do monohybrid after, and when we do dihybrid crosses after monohybrid crosses. So guys, let's move on. We only have a week to do this, so we can do a week here. And then another thing, with these dihybrid crosses, there's more going on here, guys. So it will need you to be back at the school learning, rather than me coming here on Monday and pumping you with dihybrid crosses. You understand? So let's go and do examples here. Another blood grouping, incorporating this monohybrid crosses, and then while we are learning that, we'll now take whatever. Then here's the map. I was saying that Monday we'll do blood grouping, and then we'll do a previous revision of what we did previous week, concept of inheritance, monohybrid, sex determination, sex linked. And then Tuesday we'll do question and answers with these problems, blood grouping, sex determination, and sex linked. And then Wednesday we'll do dihybrid cross, Thursday dihybrid cross, Friday question and answer. Ne? So someone was asking me about genetic lineages and pedigree last Sunday, and then I told that person we are still back. To eat too much doesn't mean you'll be full for the rest. So guys, those who are moving at the lightning speed, we will find you there. We'll find you there. Department of Basic Education said we will have to do this in a week. Yeah. And because schooling Salina, for sure, they are not only teaching you life science. So let's move on. This is the CAPS, and then from the CAPS, this will be our examination guideline. 2021, it was published 2021, so it means if we can go and check 2021, 2022, 2023, 2023 question papers, will be equipped on how they are asking their questions and how they are placing them, because you will see that when question comes, question 2, it will start with maybe difference between concept of inheritance, and then 2.2 monohybrid, 2.3 pedigree, so forth and so forth. Hence we do examination guidelines, and then we go to our textbook. So mini introduction, mini revision. We did Mendel principles, and then Mendel principles. Mendel, it's a Gregor Mendel, the father. You still remember, I even wrote wrong spellings. Gregor Mendel, as a father of genetics, this guy helped us to understand how genetics are passed from one generation to the next. And then he came up with 3 principles. The first one, which is principle of segregation. The second one, principles of dominance. And then the third one, principles of independent assortment. And we said in segregation, 2 factors, they separate. 2 factors, they separate. They separate from the parents, and then so that the gametes will have one of these 2 factors. And then the dominance, one factor is dominant over the other. So in heterozygous conditions, we will have a phenotype where the dominant one it's visible. And then independent assortment, I told you that this one we take it back to our metaphase. We'll explain this when we do dihybrid crosses. When we do dihybrid crosses, this is where independent assortment is another thing. So okay, they said have a difference or be able to differentiate between chromatin and chromosome. Someone came and said, Mr. Mpepu, the terminologies I'm not getting them, and what. And I told that person, she was a lady, by the way. I told her, what is it that you are not understanding? When learn to understand. Don't learn to cram. Don't learn to be able to recite each and every word that they said about the chromosome. When if I can come to you and ask you, ni chromosome, an accounting student can't tell us what is a chromosome, but a life science student can tell us what is a chromosome. What more are you still wanting from this? A chromosome, we have a chromosome. You don't know chromosome, you are going to have a problem. We have a chromosome, and then this chromosome is made up of chromatid, and then this chromatid, they are made up of chromatin. We talked about this, Anil. Chromatin. Then we have a gene and alleles. In this chromosome, in this chromatin, because it's a gene, because it's a DNA and then histones, we have a portion here that we will call it a gene. And then when these things matches together, or when they are at a specific locus in a chromosome, we are going to say those are the alleles. Those are the allele. One, and when now you know this, we move on. You know this, we move on. You are ready to change our gear. So you can't be doing dihybrid and pedigree. Guys, we still have pedigrees. You come to me and say, hey, Mr. Mpepu, explain to me pedigree. I'm not understanding pedigree. One cars, no. No, I'm not sure if whatever. What is it that you are testing me? That I do. I know pedigree. Guys, we still do pedigrees, but not now. First learn here. Before we can take this information and interpret it as pedigree, understand this. And then from there you'll understand, we have genes. These genes are represented as alleles, capital letter A, small letter a, up until Z. And then in these 2, we have a dominant one, we have a recessive one. The dominant one will mask the recessive one. If we have these 2 in a heterozygous condition, the phenotype will be determined by the dominant one. And then from there, we have a phenotype and genotype. You understand the genotype, it will be the gametes of that person. You might be say, we have a capital letter B, small b. These are the gametes, or these are the zygote. And this zygote is going to have a phenotype. It's going to grow into a human being. It might be black human being if this black is coding for a color. And then from there, we said we have a homozygous and heterozygous. A homozygous is when these alleles, they are the same. When these alleles, they are the same, we said we have a zygote that have the same alleles. Then homozygote. Homo, same. And then we say heterozygous, it's when we have a zygote with different alleles. Then we move on. The law of inheritance. We are moving on. The law of inheritance, but dominance, dominance, one is dominant over the other. And then you can play with words here. Here you can play with homozygous, a characteristic. They are playing with words, but whatever they are saying here, one is dominant over the other. Move on. And then monohybrid crosses. Monohybrid crosses, we talked about it, and then we said, in monohybrid crosses, those 2 alleles that we were talking about there, we are now going to cross them. Sample like this, we are going to do a spider webs, or we are going to do a Punnett square. Punnett square, we write it like this, and then Punnett square, we are writing like this. This direction, they collide, capital letter. Rule of thumb, capital letter, small letter. Now, You are going to use this information and this information to title the following problems up until the dihybrid crosses. Dihybrid crosses, it's when now you are crossing two characteristics. We are still going to do that. When at this moment, stop asking me about dihybrid question related. We will do that. Do you understand? Maybe you can just wait for me. The day I do this, you bring those questions. You wait for me. The day I do pedigrees, you bring those questions. You wait for me. Whether you are now doing human ears or human eye or whatever, you are doing response of plants. You wait for me to do the response, the human ear. You tell me those questions. For now, guys. But when you want to go far, go together. When you want to go fast, go alone. So when because you are going alone, let's move on. I don't want to dwell much on those that are pushing me to something that we are okay. The spider web and this, you are going to learn it. And when we say we are going to learn it, we are going to say A and A. Now you understand that if I come and say A and A, and then I say this will match with this will match with this, and this will match with this, and this will match with this, you understand very well what is happening. You are going to use this there, down there, dihybrid. And then from there, we understood this. Okay, we can do a Punnett square, we can do a small thing. Now we are still playing with our life science games. And then by format of representing genetic cross, we did this. And by Mendel principle of segregation, I told you that segregation, two factors separate. Then you can play with words now. And an organism possesses, or in organism there is two, or in organism cells there is two factors which separate that, so that one gamete will have one of those factors. That's the separation. And then dominant, what we call assortment, dihybrid. We'll do that when we reach dihybrid. And then types of dominant, we have a complete dominant, incomplete dominant, and complete dominant. And we said we have keywords. Complete, one allele is dominant, and then incomplete, neither or none of our alleles are dominant, and co-dominant, both of them they are dominant. It means here we'll have a dominant color. It's either black or white. If we are recessive, they have two. And then here, incomplete dominant, we will have a black multiply with a white, and then we have a color gray, an intermediate phenotype. And then co-dominant, we will have a black multiplied by a white, and then we are going to have a black and white results. It will be black, white, black, white, black, white. The leopard. It means the leopard, we have co-dominant brown and black. We don't wake up one day and see a leopard without the spot. They are the judge. Let's move on. And then genetics problem involving each of three dominants. We did the exam, we did the question papers, and also we are also going to involve this tomorrow. And tomorrow we are saying Monday, Tuesday we are going to do question papers, and then Wednesday we do dihybrid. So today you'll be fine with blood grouping. By the end of today, you should be understanding the blood grouping. We move on. We are doing a small revision here. And then by proportion and ratio of genotype and phenotype. And then we know when we are doing this gametes, we are going to have capital letter A, and then capital letter A, small letter a, and then capital letter A, small letter a, and then A and A. And then genotype, we are going to have a 1 is to 2 of this, is to 2 is to 1 of this. When you do dihybrid cross, we are going now deep into what this 1 of 1. So if you don't understand, why am I saying genotype is 1? So it's 1 of this. Capital letter, we are going to have 1. Capital letter and small letter, we are going to have 2 of them. And then once we have small letter and small letter, we're going to have 1. And then we go, this one is a proportion and ratio. So it means that phenotype, because phenotype we are dealing with dominant and recessive, we are going to check the first letter. If it's dominant, it means all of them, they are black in color. And then the 3 of them here, and then the other one, recessive, is going to be white. So we are going to have 3 is to 1. So proportion, the ratio 3 is to 1. Proportion here is 25%, 50%, and then 25%. But here it's 75% and 25%. We talked about this. And then you have to learn this. Or after that, how do I calculate the proportion? How do I calculate the ratio? Go and do question papers. guys, go and do question papers. Bring those question papers, go WhatsApp group, and then when we have them on WhatsApp group, or send them direct to me. If we haven't deal with those question in WhatsApp group, I'll forward them in the WhatsApp group. Then we'll do them when we come here. I hope that we understand that one. Then for those, my cell phone number is 0711 906168. We welcome to our class. Let's move on. And then we did sex determination Friday, sex determination and sex linked. And then we talked about that in a human karyotype. You learn about the karyotype where we have chromosomes 1 up until 23 being grouped and homologously so. And then we say 22 pairs. These two, pair number 1, it will be like this. Pair number 2, it will be like this. Pair number 3, it will be like this. So it means that we have 2 chromosomes, homologous chromosomes, that are pairs. So we are going to have 22 pairs, which will in total, it will be 44. In a zygote, in a human being, it will be 24 chromosomes. And then here it will just 22 pairs of chromosomes. And in a human are called autosomes. We talked about this. Chromosome number 1 up until chromosome number 22 pairs. Pair number 1 up until pair number 22, we call them autosomes. And then one pair of a chromosome, we call them sex chromosome or gonosome. This is pair number 23. And then I said female it's X and X, male it's X and Y. Yes, the thing I did, males have X and Y, and then female have X and X. We did this. We even went to our textbook and see how we play with words when we are explaining this. And then why do we have males why do males get affected? What? 50% chance, we have 50% chance. Sex determination, you are going to determine sex using same strategy, samono hybrid again. And then like this, and then this sex, this is sperm again. This one is a sperm. This represents a sperm cell. This represents a sperm cell after meiosis. So this sperm, there's a possibility of it to come and merge with this egg. And then if that happens, we are going to have an X and X, which this will code for a girl. Gaily. Is it alright? Girl like this, or I'm leaving something girl. Is it girl or girl? Girl. A female. Then from there you understand in karyotype I said we have 2 chromosomes like this, and then when we see in 23, when we see both of them tall and tall and homologous, same shape, same position, same shape, same height, it's X and X. And then when we see a small and long and a small, it means you have X and Y. This one is a Y chromosome, it's a father, it's a male. You still remember when I said when it comes to sex linked inheritance, why it doesn't have the location or the locus again. You still remember that. Thank you. And then they're saying we did the differentiate between sex chromosome, gonosomes and autosomes in the karyotypes of a human males and a female. You know, we did individual 1 and individual 2, where the other one, the chromosome, will be like this. They can trigger you like this. And said individual 1 and individual 2 is individual 1 a male or a female? Give us a one visible answer. You will say these two, they are not homologous chromosomes. Because they are not homologous chromosomes, we have X and Y. These two, they are homologous chromosomes, we have X and X. And then this constitute that we have a male, and this side we have a female. Karyotype number 23, the gonosomes. They can say chromosomes, give us a name of a chromosome that represents a sex chromosomes, gonosomes, or the other part that a code for every characteristics. Those are autosomes. We did that. And then representation of a genetic cross to show the inheritance of sex. How do we inherit sex? We inherit X from the father and X from the mother, and Y from the father. If you are a boy, you know you got your Y from the father, because the father is the one that determines the sex of the baby. This X can merge with this X, and then we will have a daughter. But if this Y merge with this X, we are going to have a boy. So here's the thing. You might find that this chromosome might end with this, and then we have a baby. And then this chromosome end with this chromosome, and then we have a boy. And then you find that in a family, you have a sister that looks similar to your brother. Like your relatives, they look similar. Like you have a same traits that we see from your brothers and your sisters. Ne? You can tell with this family, but same family, because for you to have a boy, one of these chromosomes, you might find that this chromosome once matched with this X, and then you have your older sister, and then you find that this X match with this another X, and then you have your older sister who is different from you as a guy, because this X match with the other X. Ne? And then sex linked, after that we did sex linked inheritance. Since we are doing this X, Y multiplied by X, X, we have the alleles that are linked in this X and Y. Where are they linked? There are those who are linked in Y for the pina hair, and then those who are linked from X, which they bring about sex disorder. An example, hemophilia and colorblind. We said hemophilia, it's a blood condition where you don't have a clot factors, your blood won't clot, ne? And then black color blindness, it's when you can differentiate between two colors, maybe example green and yellow. You see a yellow color, you say it's green. You see a green color, you might say it's yellow. And then we're saying that hemophilia, these two, they are caused by a recessive. We did this. They are caused by a recessive allele. So for you, or for a female to be suffered from a recessive allele, she will need two of those alleles. So we can say here she will have H and H, and then the guy to be affected only need a small h. And then if the mother is like this, we said here we have a carrier. She is fine, but she's a carrier of hemoglobin. Hemoglobin, we said we use HH. Color blindness, we use B unless stated otherwise. So you get me. And it's not only these two disorders that we deal with. We also deal with Duchenne muscles. We also deal with X chromosomes syndrome. And then we also done with Rett, that is caused by a dominant allele. And then now, because these are caused by a dominant allele, they might ask you that between these two, when a disorder is caused by a recessive allele, only men are in danger. But when the disorder is caused by a dominant allele, females are more into danger. Why? Because when it's caused by a recessive allele, as a male, you will only need a small h. And because females they can be a carrier, we are more into a danger because we can inherit this X and then we are affected. But when we are dealing with a dominant allele, then in a dominant allele, females are going to be more affected because when they have one capital letter, they are sick. And then when they have both capital letter, they are also affected. They will need two small capital letters X and X. And for them to get this, it means that they also have to get it from the father that is disaffected, and it's more affected. It's going to affect more onto females than to men due to this. I'm not sure if I'm explaining it right, but you will find it along the way. Let's move on. Let's not waste time. We did that. We explained that 1 hour, guys. So we can be. It can be. So from there we learned our sex linked. From there we go and learn our blood group. This is from our examination guideline, Anir. So the examination guideline started 2021. So it did 2021, 2022, and then from 2022 we also have another school year, 2022, 2023, and then from there we also had another school year, 2023, 2024. Hence now we are at 2023, 2024 school year. I'm explaining this thing here, why we have a blood grouping this like this. So okay, it's fine. From there, we're talking this side. We're talking about chromosome from 1 up until 22. I will come this side. We are doing blood grouping now, so I'll come this side. We have chromosome 1 up until chromosome 22, and then we know that 23 is our blood. It's our sex chromosome. So it means that your father. We did this example like this. Your father will have 2 chromosomes. Example. This is an example. It's not like your father only 2 chromosomes. Don't get mistaken, guys. When we say your father only 2 chromosomes and your mother only 2 chromosomes, and we are going to have 1 chromosome here, and we are going to have 1 chromosome here, and we are going to have 2 chromosomes as a zygote. We are not saying your father only 2 chromosomes. This is it is just an illustration, a visual illustration of this. This is what it is happening, illustrically, like reabonta. But what is actually happening is that we have 23 pair plus 23 pair from the mother. So it means that these 23 pair, I'm going to do like this, allow me to do this. We are going to say this chromosome, we are going to say 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, up until 23. Up until 23. And then we are also going to get another chromosome from the mother, complementing this 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, up until 23. So here's the thing. This is a cell, like this is your human cell. Ne? Hence we are saying we are dealing with a microscopic thing. If we are taking this into a day-to-day thing, we can't illustrate this. Hence we use only 2 chromosomes. But in reality, you have 23 chromosomes matching together. We'll say this side blue is from the father, red is from the mother, and then they match them. You might find that here in these 2 chromosomes, let me just do like this, because I will be like doing a new cell. Here in this chromosome, we are dealing with skin color. And then here we have allele. This allele from the father, it's capital letter B, and allele from the mother, it's small letter A. And then second chromosome, we are dealing with hips. And then let's say we are dealing with hips, and then we will have a small letter A from the father, and then you got a small letter B from their mother. And they will say, hey, shake what your mama gave you. And then with the guys, you might find that in one chromosome, we are dealing with muscles. And then you inherited a capital letter from your mother, and you inherited a capital letter from your father, from your mother. Then you know that you are big now. You have both. You are dominant now. You are homologous dominant. So you understand. Explaining this up until we reach the sex determination column here. Now, last chromosome number 23. And then this chromosome, you might say that this one will be XY multiplied by XX from your mother. So let's say in your father, this will be 2 chromosomes, man. This will be 2 chromosomes, and then let's say maybe you got X from your mother, and then Y you got Y from your father. We'll write it like let's just use X and X, so that the colors will not fight, and we won't have a color mix. Because Y will have to write it at the other side. Okay, let's say like this. Now we are at the last chromosome, sex determination. And when we come to these cells, I know this is a 1 cell. You start as a 1 cell, my guy. You started as a 1 cell. Before you can be a multiple cell, you started as a 1 single cell. This cell that I'm talking, this was you at some point. And then we came to this number 23 at our karyotype. Number 23, we saw that you inherited X from your father, and you inherited X from your mother. So you are going to be a daughter. How do we write a daughter? Daughter. I'm not sure. Do we write a daughter like this? So from there, we go back now and said, okay, we are learning you. Okay, it's a girl. We are right. She's a girl. We found it here. And then chromosome number mang, chromosome number 20, chromosome number 22, chromosome number 20 what, chromosome pair number mang. And then somewhere in these chromosomes, we also have chromosomes that are pairing for blood group. It's not another cell. You are a cell when. We also have these 2 chromosomes that they are now heading for a blood grouping. When we do a random and independent assortment, what we did an independent, let's come here and explain independent assortment when we're explaining this. We did separation, they will separate. We did dominant, one will be dominant, one will be from the father. And then we do assortment. Assortment is that when we have this when in your father, when this is happening inside your father's cell, for them to separate and give you this blue, we had a blue, maybe let's just say an example, this one was your father, This one was your father, Your father had blue this side, and then he had this before you can divide them. Ne? Before we can go meiosis. Before meiosis, these chromosomes were mixed, and then they had to regroup at the equator so that they will divide. And then you find that this one, he got it from his grandmother, and then this one he got it from the grandfather. So now he's inheriting them to you. Then here's what is going with as independent assortment. What is happening is that you might find that this chromosome from the grandmother is staying like this, and then when you come to pair number 2, which one is okay? We are using blue. Pair number 2, you find them staying like this. Pair number 3, pair number 4, I'll come and fill in the one from the father, the one from the grandmother, and then one from the father. You find that they are randomly placed at equator. Ne? Independent assortment, they are randomly. So it means that when this gamete, your father's cell, your sperm, sapapao, when it's going to divide and form these two these two cells. The other one having this part and another one having this part. And up until the chromosome number 23, where we will have X and Y. Hence we said 50% of your father's sperm, it's Y, and 50% of your father's sperm, it's X. So when these two come and join to you, yes, we will have this blue representing from your father, but you'll find that in this blue, you got it from your father, but the father sent it from the mother. And then this one, yes, it's from the father, but the father, he inherited it from the mother. Here's the thing: father, mother, and then to your father again. This is your father, and then this is you. And then we say mother, she passed this, father, she passed this. He passed this. And then when they're two, they divide down here. After dividing, now they are randomly assorted. It means that here you will also have same thing with your mother. Your mother, she have a possibility of giving you the one that she inherited from her mother, and giving you the one that she inherited from her mother, and give you the one that she inherited from her father. Something like this, it's going to happen. Assortment. When we talk about assortment, then we'll talk about dihybrid crosses, when these two chromosomes, whether they are brown skin color, it doesn't affect the other two chromosomes. When if you have brown eyes, or when you have a brown skin, doesn't affect you having a curly hair. Understand that? I want you to understand. You get that? This is a complex thing. There's no cramming here. The moment you find yourself cramming this thing, you are just going to. Hence, you will find yourself in examination singing mitosis, wangena ku mitosis, mitosis, wasni ku mitosis, because you no longer understand what was happening when you just got the rhymes. But if you understood, no man, when this haploid cell infused with this haploid ovum, the pairs that we are going to have 46 chromosomes, and in these 46 chromosomes, each and every pair is responsible for something in my life or in my body. Whether that will be my shoe size, my fingernails, my eyebrows, my lashes, my hair color, whether they are going to be curly, it is controlled there. So now they say we have two types. We have those that are controlling everything rather than sex. We call them autosomes. And those who are controlling something else other than something else, we call them sex chromosomes. Those are controlling sex characteristics. A man, you are a man because you have a reproductive whether Caster Semenya. We can't say Caster Semenya is a man, because the sex chromosomes, she's housing XX. But what we see physically, it's what she inherited from the father. It means that her father's gametes or alleles, they were dominant. Where now you have someone now in your class, he or she or he is a gay. Let's just put it like that. He's a gay. And then you might say, Mr. Pip, is this person acting or he is like this? Explain it genetically. It means that the chromosomes that he inherited, Yes, at the point number 26, he inherited the one from the father. But the other chromosomes, maybe the mother gave that person the chromosome that had a more dominant alleles. Voice, female is dominant. Figures, that person you find is a boy, but unali figure. Unali mara, unali matswele, the face, it's like, you understand? If it's that person, it's a girl, you find that she's a lesbian. It's a guy, he can even rob you. Even the voice, even the beard, even everything, but she's a girl. Now you understand, or it means that something that happened is not a mistake. There was not a mistake. It's just in a pair number 23. She inherited an X from the father. But the other chromosomes, the other 22 chromosomes, they were having a dominant allele from the father. And it is in this sperm cell here having an X, and in this X we are having other chromosomes that we can see that due to random arrangement at the equator, now it's going with an X, but with you have X here, you have an X that is going to determine that our baby is going to be a daughter, but that X, it's moving, or maturokisi, it's hacking maturokisi of things that are going to be main thing. The person now gay. You understand the trauma? From here, this is what we are talking about. When I explain this, you can explain go high. If maybe your relative, maybe your brother is a gay, and then go high when you come disown, demon. No. Go and explain to them genetics. Go and say, Mama, you know there's something called genetics. You know where no. So then we are going to do genetic engineering. Because luckily, so I can remove my DNA. Now you understand. I can come and choose these DNAs and take these genes and place them on another cell, on straight to the ovum, and we are going to force that ovum to have a specific person. If maybe we want a boy, we are going to say cells. We are going maybe to study the cells. Maybe we find the difference that we have cells that have short sperm cells that have a short chromosome tail, and then we have a sperm cells that have a long sperm cell and a big tail, and then we infuse this with another ovum, we infuse this with another ovum, and then we find that this ovum is a boy and then this ovum is a girl. Then we conclude that the one with small tail, they are housing X, and the Y with long tail, they are housing Y. So we can take, or we can harvest these sperm cells and go and sex the female part here, and then this female part, she's going to bear the bulls. Those who are in Greek now, you want bulls or you want bulldogs that they are males, then you can use this genetic thing. Maybe you want color, you want puppies white color, you use these genetic things. So now we are at blood grouping. In blood grouping, why do we now in blood grouping we see these three dots, and then they're saying a different blood groups are results of multiple alleles? Let's explain this in these 2 minutes. We are going to say in one of these chromosomes, we are going to have chromosomes like this. In one of these chromosomes, we are going to have chromosomes like this, and those chromosomes, they will have a specific locus here, or a position housing a specific locus. But in this question, in this chapter or this topic, they will be housing a blood group. They will be housing a blood group or the blood allele. And then in this blood allele, there's three possibilities. It's either there is a chromosome with a allele A, or allele B, or nothing. This is what I used to say. I used to say nothing. These are commas. I used to say this one is an allele. Although some they will say it's O and what. But this one, it's nothing. It's like you have this allele, and then there is A, or there is this allele, there is B, or there's a possibility of having another chromosome that is having nothing. So here's the thing. We write them like this. We have A with capital letter A, B with capital letter B, and I with nothing. And this I is recessive over this. Here's an example. If we find a person having a group A and a small letter I, it means that person in his locus, the locus is filled with A blood, and then we are going to say this person is a blood group A. We have 4 types of blood groups, and then another thing we are going to say, if we are going to have a let me have a space. Let me write this. I said 2 minutes. And then we are going to say, for example, let's say you have a I, B with small letter I. Because this one is a recessive, or this one doesn't affect much the results because there is nothing. It means this person is going to be group B. And then A, B, and then you might think that if we're going to match A and B, we are going to have C. No. This one, they are complete dominant and we have co-dominant. What do we mean by that? When we have these two, rather than having another intermediate phenotype, they are going to share the same phenotype, and then we are going to say this person only blood group A and B. And then when we have nothing, Mr. Mpepu, when we have two chromosomes that in those locus there is nothing. In those locus there is nothing. We are going to have a blood type O. Nothingness. Let's write it down here. We are saying that we will have two chromosomes. Another thing, don't you mistake in this. We will have two chromosomes. It's either you'll get one from your mother or one from your father. Because the one that you are going to get from your father, it will be dependent of the two. And then these two, it was from another two. And then these two, it was from it's when you do the pedigree and do the lineage. That's where you will be now studying this. Because we're not to have this chromosome and this chromosome. This chromosome, we got it from your mother. Your mother, she might give you this or this, depending on this coming from and this coming from. You understand? So when now you might have a one chromosome from your mother that it was inherited from her grandfather, that it was inherited from their grandfather. So it means that fatherly chromosomes, it is with you now. And then from the father, you carry the one chromosome that is going with an X. Here we are going to be a girl, but the girl that is a boy. Because now the other chromosomes that they are making you inherited them from your mother, that she inherited them from her father, that the father inherited them from his father. Like you have this fatherly characteristic that was passed but recessive, and now they are matching with. Be able to explain that. When you are a life science student and you are doing this, be able to explain that. Be able to explain that we have three possibilities in that chromosome. It's chromosome number what? I'm not sure. But in that chromosome, we have three possibilities of us having something in that locus. In that locus, there's a possibility of that locus to have A, there's a possibility of that locus to have B, and there's a possibility of that locus to have nothing, which is O. And then because the A is dominant over O, when we have this matched with small letter I, we are going to have A blood. And when this matched with small letter I, we are going to have a B blood. When we have I, small letter I, we are going to have an O blood. But when we have these two, when we have A and B, we are going to have blood group A and B, because they are what? They are co-dominant. They share. They are both dominant. My daughter, man. Find this life science. They are both dominant. Co-dominant, black and white, we have black and white. We don't have a new color. So that was it with blood. And then when we cross blood, how do we cross blood? We are just using these eyes and eyes. For example, we might say an heterozygous an A person, an heterozygous person of blood group A. It means it's heterozygous. They have A and small letter A. The person is crossed with a person with A and B. It means you have A and B, and then you cross them. This and this, it will be A capital letter and A capital letter. And since these two, they are both dominant, it means the phenotype will be A group. You get me? So even if we have something like this, because this one is dominant over this, we are going to have a B group. So B group, it can be represented by this, or it can be represented by this. Heterozygous, homozygous. So, by genetic problems involving the inheritance of blood group, then we have to do these problems. Ne? Then from there we are done. We can move to dihybrid crosses next Wednesday. Ne? Thank you for tuning in, guys. And then we move on genetics, those who will be like Rinyaka notes and what you have is what do we call this, mind the gap. And with mind the gap, you have everything that you need to know, unless maybe you want 100 out of 100. You might now search and go and search another thing, but mind the gap, you have mind the gap, you have textbook. Mind the gap, they told you about the gene. We talked about alleles, genotype, phenotype, dominant, recessive, heterozygous, homozygous, and then monohybrid, complete dominant, incomplete dominant, and co-dominant. And then you know the keywords here. We are going to have one allele that is dominant over the other. Here we are going to have non-allele or neither allele, and we are also going to have an intermediate phenotype. Co-dominant, we are going to have both of them. And then example, it's also in blood. We also find this in blood. And then black mixed with white, we are going to have black and white. Here we are going to have gray if we have to mix those colors. And the multiple alleles, there are more than 2 alternative forms of a gene at a same locus. They are not saying, as that example they use blood, they are not saying that in a blood we have 2 alternative forms of a gene at a same locus. They are not saying at a same locus we have 2 types of gene, like we have A and B. No, they are just saying that in this locus we have more than 2 possibilities. It can 1 A or B. Then the third possibility is when we have nothing, we have multiple alleles now. And then this one, it will be code for the O. This one called for B, then code for A. Then sex-linked characteristics, karyotype. This is what we did. And then are these homologous? If the answer is yes, it means it's X and X. If they were heterozygous, they will be X and Y. And then they will be like this, and then they will be like this, or they will be like this and like this. This one after intermediate, after replication. Then you have to learn this one, big X female with small one male. And then we'll go and do cloning, we'll leave it, genetic modification, you leave it, human genome, you leave it. And then from there we can now move to our questions. Our question number 1, the allele that is not expressed in the phenotype when found in a heterozygous condition, not expressed, recessive, must. And then different forms of gene, different forms. These are the different forms of a genes. We have different forms of genes which occur at a same locus. These are the keywords for something. On a homologous chromosome, we are talking about an allele. You see how we get it. And then number 3, a sex-linked condition. A sex-linked condition. Here we are talking about sex-linked condition where blood fails to clot properly. Hemophilia. Number C, we talked about this. And then number 4, the pair of chromosomes in a diploid organism that have the same size, same shape, and control the same set of characteristics. We are talking about homologous. Homologous are the pair of chromosomes that have the same size, same shape, and control the same set of characteristics. And then the physical and the functional expression of a gene. Physically and functional expression of a gene. Here we're talking about when we have a gene that is like this. The physically, we are going to talk about phenotype. Where's the phenotype? Number G. These are the physically or functional expression of a gene. Then we can go. This is the answer. Then we move on from the mind the gap. Mind the gap. We can write something here 2 minutes. Then 2 minutes we go and you can go to our textbook, and then from there we did an example. We can say you understand now there's a cross that we are dealing with. We are also going to use these crosses on a dihybrid, so you'll have to be able to understand that if I can cross like this and if I can cross like this, I'm still crossing the same thing but different method. And in this one will be A and A, small letter a, and then this one capital letter A, small letter a, and then A. You'll know that this is what we are also going to use that side again, and then we understand that it's the same as when we present this. We are going to do like this. We are going to do like this, sex determination, and then sex linked. You'll be like this. We don't touch the X. we don't touch the X, and then what we call this blood typing, we are going to do like this. And then heterozygous B group, and with heterozygous we have 4 types of groups. We are going to do them, and then we know that this one will cross with this one, also this one, this one, this one, this one, this one, and then we'll show capital letter B, a small letter capital letter A. Here we start with blood group, we start with A, Anir. Blood group, we start with A, then we'll say A, and then we'll say B, and then we'll say B, I, and then we'll say 1A, and then I, and then I, then I. So the genotype will be like this, and then it will be 1 is to 1 is to 1 is to 1. So you'll be phenotype, you'll be AB, and then you'll be B, and then you'll be A, and then you'll be O, and then this is the outcome, phenotype. So we move on to our classwork, and then to our textbook. And then with our textbook, I gave you this exercise to say go and do it, and those who did it, thank you. Those who didn't do it, we are moving on. You'll do the next ones, but read the following extract on the cystic fibrosis and answer the question that follows. And then you will read the cystic fibrosis, and then the questions that we can screen. It's a chromosome number 7. It's caused by a recessive allele, and then 1 of 20 what we can move on. And then question number 3.1, explain why a cystic fibrosis is not a sex-linked chromosome, because it's taking place at chromosome number 7, and chromosome number 7 is not a sex-linked, 2 marks. And then number 3.2, use a genetic cross to show what percentage of children will be affected if one parent is heterozygous, then heterozygous, capital letter B, small letter b, and the other is a homozygous. The recessive allele is represented as B. Use the genetic cross to show them affected if one parent is heterozygous and the other one is homozygous normal. Okay, homozygous normal, it means will be like this, because it's caused by recessive allele. It means for you to be normal, you will be opposite. Let's do like this. You are heterozygous B, small letter b multiplied by a normal capital letter B, and very show us the percentage. Show us the percentage. You are going to cross this, and then this and this. It's going to be B, and this and this. You are going to be B and B, and this and this. You are going to be B, and then this and this. You are going to be B, and then genotype, it will be 2 is to 2, and then phenotype, it will be 100% what? 100% normal, because it's caused by a recessive allele. And then for someone who's caused by recessive allele, you will need small recessive for it to kick. So the percentage, there is 0%. Okay, you write the compulsory one, that there is a 0% chance of children to be affected. You get your 6, and then you use a Punnett square. You know how to use a Punnett square. You're also going to use this Punnett square when we do our dihybrid. We are going to do things there. Then it will be B small letter B, B small letter B, capital letter B, then it will be B, then it will be B, small letter B, and then here B, and then B, small letter B, and then gametes, and then phenotype. Number 2, 3.3. Maggie and William want to start a family, but Maggie's brother had cystic fibrosis. It means on the brotherly side. Yo, I didn't do a correction, but now I see that. Man, this one is a sex. Oh, it's not a sex linked. Oh, as kiss. I thought because I was reading a sex linked here, we have to link this. No, it's not a sex linked. See another. I thought maybe these questions were dealing with sex linked, so we had to link them. Hence now they are also saying that Maggie and William want to start a family, but Maggie's brother had a cystic fibrosis. She doesn't want her own child to suffer as her brother did. Maggie and William decided to visit a genetic counselor. You see, You can also be a genetic counselor when you grow up. If this topic fascinates you, you can also be a genetic counselor. I've explained how this may help Maggie and William in their decision. This can help them to determine whether to have a child or not to have a child. We can use the Maggie brother, but this is the only two that they want, and to explain how this may help Maggie. So it will help them to make their choices whether they want to have a baby or not, depending on their possibilities of them to be to have a cystic fibrosis, given that she has a brother that is suffering. So it means that here as a female, since it's not a sex linked, it means here as a female, she might be suffer. There's a possibility of her to be suffering or to be like this, since she's normal and it's caused by a recessive. And the father that side and the William this side, there's a possibility of William to be like this. And if William is like this, they can have a baby that is affected. But if William it's completely normal, dominant, and then there's a possibility of them to not have a child that is cystic fibrosis. You get me? So don't be confused, guys. When we deal with sex linked, that's where we linked our thing. But here we are dealing with chromosome number 7. And they said, explain why cystic is not a sex linked. Why are we not linking this thing here? Hence I said I like this. You really remember, ne? I was talking about this confusion that they might bring to confuse us. Then from there, we move, even our textbook, move straight to blood type. And then blood type, the genetic crosses dealt with thus far involve the two alleles of a gene, e.g. T or small letter t for tall, R or W. Sometimes a characteristic is, however, controlled by more than two alleles. Blood type or blood grouping is an example of such characteristics. This characteristics is being controlled by more than two alleles. There are 4 blood group types in human. It's either A, B, AB, co-dominant, or O. Dominant, complete dominant, co-dominant, and then this one will be dominant also, will be complete dominant, because with two under complete dominant, when we have 2 recessive alleles together, that's when the phenotype is going to be for those recessive. And then these phenotypes are controlled by 3 alleles, but each person still inherit 2 alleles. That's what I was talking about. Or you are going to have 2 alleles, because the alleles will be shared from your father's father from 2. But if there's a possibility of you to have a chromosome that have nothing, and then when you inherit another chromosome from your mother that have nothing, you are going to be type O. And then it is very important to know how to name or write down these 3 alleles, as they are very specific to blood group types, namely blood group type A, blood group type B, or blood group type O. A is co-dominant to B, whereas I is recessive to both. We know that. And then the genotypes for each blood group can then be specified as follows: genotype and blood group. We did this, and then since this one is a dominant, and then if we have capital letter and capital letter, it's going to be A. And then if we have capital letter and nothing, we are going to be A. And then if we have capital letter B, capital letter nothing, because it's dominant, B. And then when we have both of them, it's A and B. When we have nothing, it's O. Ne? So if we can say a homologous A blood, a heterozygous A blood, a homologous, a co-dominant group, a blood group which is co-dominant was crossed with, co-dominant. Do you understand? A blood group type O. You have to learn this. A blood group B, homozygous. A heterozygous blood group B, a homozygous blood group O. Yeah? What's the judge? A homologous blood group B crossed with homologous A. So all of them 100% they're going to be A, B. Hence I did the the explanation why we explain this as a father. And I'm not sure if I did it on our previous group, on our previous video, because I did a mistake with my previous video. But I did say here we can use to explain the father and then to explain the mother. If the person is your mother or the person is, I'm not sure if I'm repeating this. Am I? No. Here I also say that let's move on. You'll see use of blood groups in paternity testing. This is what I was referring to. But the blood groups of a mother, a possible father and child must be compared. If the blood groups of the adults do not correspond or match the child's group, then this man is not the father. If the blood groups of adults correspond or match the blood group, then there is a possibility that the man is the father, and other tests need to be done, as other men may have the same blood group. Only DNA proofing can be conclusive, as it looks at the similarities between the nucleotides in the DNA of the father and the child. Each DNA profile is unique to a individual. 50% of DNA fragments bends, bars, and divide from. This is another information. But let's talk about this. My time elapsed, and I thought maybe I talked about this, but let's talk about this. We are saying that if you have as a father, and then you be like this, we know that your blood group is AB, and then you are going to be with a father, like with a mother like this, and then they are going to say a baby, she is type O. A baby, she is type O. Is this the father to this child? Yes or no? 2 seconds. Answer no. Answer no. Why? Explain it. Why are we saying no? Because for this child to be O here, she has to be small letter I and small letter I. And since the father can't give the baby small letter I, it means that this baby got the other I from another father, maybe neighbor or something, but ngwana waso akamumti. Hence the ancestors, or what they will say, hence people will say, give me blood. Give me blood. Let's go and do tikotamadi. Yes, they might be referring to DNA, but also the one that is first is blood. And we use blood to exclude someone as a father, not to determine that this person is 100% father to that child. You get me? We use it to exclude a father. For example, we can say this guy here is not the father. So who's the father? We don't know. And then another example is when we have something like this, multiply with something like this, and then they are saying the father, they are saying the child here. Let's do like this, and then they said here the baby is like this. Is this father here, since the father is a type A blood, is this guy the father to this child? The answer is yes, because we might find that this B, she got it from the mother, and the I, she got it from the father. Although this one is A blood and this one is AB and this one is B blood. And another thing, why don't we use this to conclude the paternity of the child? It's because we have more people that have same blood groups in this world. It means someone can have like this A blood group, someone can be like this and an A blood group, because we only have these possibilities for people to be A group. It means that most of us, we are like this. It's like when we are determining sex, because we only have two possibilities of a male and a female, a male and a female, and then we say, is this a father? Because it's a male and the child is a girl, is this a father? What are you going to say? Are you going to say no, it's not a father because she doesn't have an X, she doesn't have an Y? Yes, it's a father because there's a possibility of this and this to match. Hence, even this test theory, your father is like this and then your mother is like this, and then when now you're like this, are you the son or are you the offspring of this man? It's not reliable. Yes, you are the offspring of that, but if you become a chimpanzee, if you look like a chimpanzee, you will know that those chromosomes that this person is revealing, he or she didn't get them from the father. So with blood, it's like that. We can use blood estimate or to determine or to exclude someone as a father. When at your home we have a father like this, I did this, we have father like this multiplied by mother which type O anir, and then with these two where now you are type B, and then we say, was that your father because your father is AB and your mother she's O? Are you the child of that boy? We might say yes, you are the child of this man. And another thing, when we do the blood transfusion, it's also important. They might ask you, why is it important for a blood group to be determined before or prior the blood transfusion? This is the case. Now you will do blood tomorrow. Now let's do this. I will explain this tomorrow when we do the exam. I will start by explaining this thing here, the blood. Ne? We have a lot to explain here, and then don't want you to end up not understanding the main thing that we have to understand in blood, because blood it also have this thing. The blood produce antibodies. The blood is made up of components, white blood cell. You don't have to know that, right? That's what you don't need. When you see someone now starting to talk about, hey, your blood is made up of plasma and red blood cells, you will learn that university. Here you just have to learn that we have 4 types of blood. A, B. Hey, there's a antigen, there's what. Just know that when we mix we don't mix blood. Where's another information? And my time is up. We don't mix blood. For example, when you have a group and then this means that let me also expand this class. This means that you are red blood cells. I didn't want to explain this, but I feel like I have to. It means that your red blood cells, they have an antigen for A again. And then it means that in this blood of yours, this red liquid, what is blood? The blood is a red liquid. In this blood of yours, you will have red blood cells with these antigens, and you will also have antibodies that will match, that will be opposite to this antigen. And then when you have B blood, you are going to have a red blood cells with a B antigen. Hence we said here it's nothing. You are going to have a red blood cells with B antigen and antibodies that are for A. So it means that if now you take this blood and you pour it to someone having a blood group O, it means that we are going to have these antibodies fighting these red blood cells. Hence we don't encourage you guys to do but the Bluetooth heroin thing. The Bluetooth, you know it. If you don't know, let's move on. And then when you have nothing, it means that your red blood cells, they don't have neither the B or the A antigen. It is simple like so. It means that in its blood thing, in its red blood cell, we are going to find antigens for both blood group A or blood group B. And then another example is when we have this A and B. It means because we have A and B, all of them, they are dominant, guys. All of them, they are dominant, so they are co-dominant. It means that we will have a red blood cells with both antigen A and antigen B, because it's A and B, and self-explanatory. A, B. It means that, and then in its blood it's made out of red blood cells, the one that I'm talking about here, and then it's made out of white blood cells, masholambili, and then it's made up of the platelets, and then it's made up of a plasma. So in this plasma, that's where we'll find these antibodies. And then here at A and B, it means we don't have antibodies, no antibodies. So it means that whether you can take this A blood and pour them into someone that is having AB blood, will be fine, because nothing is going to kill that red blood cells. Also, if we add this 0 to the same thing, and same as, but problem here, when we add this blood group, we are going to fight here, because this blood here, it will contain a something that we call them the antibodies, that they will come and join here. You did this in grade 10, grade 11, immunology. When you did immunology, they will come and block here and cause the clot, and then in your blood, and you will die. So that's it. That's it with blood, guys. That's it with blood. So in this process, we are determining, or we are finding a probability. If we are saying where you have a chromosome that is going to encode for this, and then you are being crossed with someone who have a chromosome that they are going to cross for this. You have one chromosome that have B, you have chromosome that have nothing, and we do our Punnett square. If we want to do our Punnett square, and then you say here A small, then B small, and then you will have A, B, something like that. And so here you know that when these two chromosomes form, we are going to they are going to produce, or they are going to code for a red blood cells that have both A antigen and B antigen. Actually, when we do this, we are doing this as a zygote level. We are still studying a zygote. So it means that when we have A and B, they are going to code for blood cells like this, and in these blood cells, they are not going to produce neither these antibodies. You get me? So when we have I and I, they are going to code for a red blood cells that have nothing on its side, on its outside. Ne? And then when we have A, it's A only. When you have B, we have A only. When you have B, we have B only. Vice versa. Blood group B, antibodies A. Blood group, something like that. Ne? Creme that. And then number 11, sorry guys to waste your time. Number 11, they say, question here, a man and a woman both have a blood group B. Ne? Use a genetic cross to show, and then B, you know, it can be homozygous and or heterozygous, either the two. But use a genetic cross to show how it is possible for them to have a child with blood group O. So it means for them to have a blood group O, now you understand. Now they are pulling you to more understanding. Now you are now graduate. Now you are given less information. You know, for that person to have a blood group O, it means that they have to get a small I. It means our parents, because they are both blood group B, we are going to say they are going to be B small letter, so that this small letter will be given to our child, that they will have O, because we are show how it is possible. And then we are also going to do like this and I, and then crossing this and this, it's going to give us B, this and this, it's going to give us B and I, this and this and this is going to give us B, going to B and I, and this it's going to give us I and I. Ne? And then this is the possibilities that they want. Then how do you represent it? You represent it like this. P1, F1, you get your mark. Meiosis, fertilization, and then genotype, blood group B, and then genotype, you do like this, you show whenever that you understand that you want to reach a point where you have I, you do like this, you are fine, and then you do your crossing, you are fine, and then we want to see 3 groups B and 1 blood group B. Now, if they said show us a chance, it will be 75% and 25%. Ne? So, or you can do a Punnett square. You can do a Punnett square where this B is going to match with this B, yes, B, and then this one is going that direction, and then something like that. And then you do like this, you get your mark. Ne? Then activity 5. Tomorrow we'll be doing questions. So guys, let's go and do activity 5, and then also bring another questions from another question papers that we can do. I've already wasted your time, guys. And then number 1, now I'm coming. So guys, thank you. Let's do activity number 5, and then after activity number 5 we will come and do activity 5 tomorrow together with those questions. So that is from me, guys. Do a screenshot. After doing a screenshot, you can do these activities. Thank you, guys. Until we meet again, peace. Don't forget to subscribe, guys, to like and to share. We are brought to you by African Transcribers. Thank you, guys. Thank you.